students. In this lecture I am going to explain a concept called synapse and neuromuscular junction. Synapse, it is the functional connection between a neuron and the second cell. So we could also say the junction between two successive neurons, but here the first cell is always a neuron, but second cell, it may be a neuron or it may be a muscle cell or it may be a cell from the gland. For example, if you take in the CNS, in the central nervous system, the first cell is always neuron, whereas the second cell is also neuron. So this is in the CNS, in the central nervous system. So in CNS, the junction, the synapse is between the two successive neurons, whereas in other parts of our body, like in the muscles, in glands, the first cell is the neuron and the second cell may be muscle, and first cell is always neuron, second cell may be a gland, the cell from the gland. If you take neuron synapse, so in this lecture I am going to speak about this neuron synapse, but the synapse between neuron and muscle, we could call it as neuromuscular junction. It is a neuromuscular junction. In neuron synapse, there are three types of synapse, so based on its structure. So if the synapse is between the axon of one neuron and the dendrite of another neuron, such kind of synapse is called as axodendric synapse. So this one is axodendric synapse. This is the first type. Whereas if the synapse is between the axon of one neuron and between the cell body or cyton of another neuron, so this type of neurons is called as axosomatic neuron. This type of synapse is called as axosomatic synapse. Right, this is the second type. And if the synapse or junction is between the axon and between the axon of another neuron, this type of synapse is called as axoaxonic synapse. But in our body, most of the synapse in our CNS or in our central nervous system are axodendric synapse. And as usual, you know, the neuron, it has three major regions you could see in the neuron: the cell body, the dendrites and axon. If you take, this is one neuron. In neuron synapse, the synapse is always formed between the axon, which means between the synaptic knob of one neuron and dendrite of another neuron. These are the dendrites of another neuron. This is like this. So this is the cell body, it is cytoplasm, and here you could see many dendrites. So this is again axon. long unbranched cytoplasmic process. So again here you could see the synaptic knobs like this. In our body, the neurons, especially in our central nervous system, the neurons are interconnected. So this portion is the synapse. This is one neuron, this is another neuron. So this is how the synapse is formed. First let me explain the types of synapse based on its function, means how they transmit the impulses. There are two types of synapse. One is electrical synapse and the second one is chemical synapse. So there are certain points that you have to remember about these electrical synapse. Here, pre and post synaptic membranes. Or in close proximity. Means area of contact. Their area of contact has less electrical resistivity and the next point that you have to remember, so these pre and post synaptic membranes in electrical synapse, they are generally joined with the help of gap junctions. Here you could see the presence of gap junctions. And these gap junctions may be measured about only 2 nanometer. Only 2 nanometer. And here in the electrical synapse, so the gap junctions, they are generally connected with an array of 12 proteins or an array of 12 hexagonal proteins. So these array of 12 hexagonal proteins which connect the pre and post synaptic membranes or which form the gap junction are called as connexins. So through these connexins means there are 12 proteins. For example, if we take the pre and post synaptic membrane, if you take this one is the presynaptic membrane, this is the presynaptic membrane, and this one is the post synaptic membrane. So there is a very narrow space. Generally the cleft or gap is absent. This one is the post synaptic membrane. Presynaptic, and this one is the post synaptic membrane. Here you could see the gap junctions like this. So these connexin protein, for example, this is one connexin protein, this is the second connexin protein, this one is the third, this one is the fourth, here is the fifth, here is the sixth. So there are 6 connexin proteins in presynaptic membrane and thus again 6 connexin proteins in post synaptic membrane. So these connexin proteins, here 6, here 6, they together form 12 proteins. These 12 proteins, they form a water filled channels through which the ions move from presynaptic to postsynaptic membrane. And in electrical synapse, the transmission of impulses is rather fast because there is very less space. Generally the synaptic cleft or gap is absent. That is one reason. And here the conduction occurs through ions, which is very fast when compared to the chemical synapse. These gap junctions you could see in especially in cardiac muscles. in smooth muscles like uterus. So here in cardiac muscles, these gap junctions, they helps in the contraction of entire myocardium as a unit, and just like in uterus also, the gap junctions helps in the contraction of a uterus, especially during the labor or during delivery of the baby, that make the uterus to contract more vigorously. And these you could also see in the lens of eye, right? And this is about the electrical synapse. Next, the second type of synapse is the chemical synapse. Here in chemical synapse, the transmission of impulses from one neuron to the another neuron occur with the help of chemicals called as neurotransmitters. Example for neurotransmitters is acetylcholine. There are so many other examples that I will explain in some other lecture. But here in the chemical synapse, the pre and post synaptic membranes, they are also somehow in close proximity, but there is a synaptic cleft in between this pre and post synaptic membrane. But here between the pre and post synaptic membrane, there are certain proteins called as cell adhesin molecules. CAMs, we could say it as. These CAMs means cell adhesin molecules. These cell adhesin molecules that helps to keep these pre and post synaptic membranes in close proximity. But if you magnify this area of chemical synapse, like you could see the structures. This is the axon terminal, this one is the axon terminal, and this is the synaptic knob. This is the axon terminal, this one is the synaptic knob, and this membrane is the presynaptic membrane. In case of neuron synapse, the next postsynaptic membrane must be of dendron or dendrite membrane. So this one is the postsynaptic membrane. So this postsynaptic membrane has many receptors like this, which helps to receive the neurotransmitter. So this one is the postsynaptic membrane. And the area between this presynaptic and postsynaptic membrane, the gap, this area or this gap is called as synaptic cleft. So a synapse has these three areas, that is presynaptic membrane, synaptic cleft, and postsynaptic membrane. But here in the synaptic knob, you could see mainly a cell organelle, like many mitochondria are found in this synaptic knob, and it also has many minute vesicles. So these vesicles, membrane bound vesicles, are called as synaptic vesicles. Right. And in these regions, like in the axon terminal and as well as in the synaptic knob, we could see certain voltage gated channels or calcium ion channels are found here. Here, how these neurotransmitters which are found inside the synaptic vesicles helps in the transmission of impulses from one neuron to another neuron is most important thing. When the electric impulses or when action potential is released or when it reaches to the axon terminal, when the action potential reaches to the axon terminal, in response to the action potential, the voltage gated channels will open. So these voltage gated channels will open, and that causes the influx of calcium ions. So first point in release of a neurotransmitter, action potential when reaches to axon terminals, this stimulate or causes the opening of voltage gated channels. When these voltage gated channels open, that causes the influx of calcium ions. So these calcium ions, when enter inside this synaptic knob, they bind with a protein which is believed to be synaptotegmin. So these calcium ions bind with proteins, certain proteins. These proteins are believed to be synaptotegmins. So it forms calcium synaptotegmin complex, or you could say calcium protein complex. This calcium protein complex or this calcium synaptotegmin complex that helps or stimulate the binding of these synaptic vesicles to the presynaptic membrane, which means the first docking means attachment and then fusion. After docking, it fuses with the presynaptic membrane, and then it is followed by exocytosis. Through exocytosis, these synaptic vesicles release the neurotransmitters. These are the neurotransmitters. These neurotransmitters will bind to the receptors. These are receptors present in postsynaptic membrane. So after the influx of calcium ions, it forms calcium protein complex. This calcium protein complex stimulate docking, which means attachment of synaptic vesicles, and then fusion of synaptic vesicle with presynaptic membrane, and then exocytosis of these synaptic vesicles that finally releases neurotransmitter chemicals. As this neurotransmitter binds to the postsynaptic membrane receptors, that again stimulate the opening of ion gated channels. Opening of ion gated channels are gates. So here in this case, the chemical which is called neurotransmitter, it is regulating the opening of the gated channels. Hence this synapse is called chemical synapse. So students, this is about the synapse, chemical and even electrical synapse we have discussed in this lecture. So in the next lecture, I am going to explain the neuromuscular junction and then the further topics will deal in detail.